Okay. Welcome to this week's seminar. We would like to start off with our land acknowledgement. Iowa State University aspires to be the best land-grant university at creating a welcoming and inclusive environment where diverse individuals can succeed and thrive. As a land-grant institution, we are committed to the caretaking of this land and would like to begin this event by acknowledging those who have previously taken care of the land on which we gather. Before this site became Iowa State University, it was the ancestral lands and territory of the Biogeochemical or Iowa Nation. The United States obtained this land from the Meskwaki and Sauk nations in the Treaty of 1842. We wish to recognize our obligations to this land and to the people who took care of it, as well as to the 17,000 Native people who live in Iowa today. Now I would like to introduce Dr. Huang. Dr. Wenjuan Huang is a research scientist at Dr. Stephen J. Hall's lab in the Department of Ecology, Evolution, and Organismal Biology at Iowa State University. She received a PhD in ecosystem ecology from the Chinese Academy of Sciences and a bachelor's of science in ecology from China Agricultural University. She was a postdoc in Dr. Hall's lab when she came to ISU in 2016. At ISU, her research has focused on soil carbon dynamics and nutrient cycling, especially nitrogen and phosphorus, and the response to climate change in the natural and managed ecosystems. Her recent work is to understand how biological and geochemical factors interact to influence the composition and persistence of soil organic matter.
Seems more is the laser at the same time. Thank you for Marshall's invitation. This is my great honor to stand here to show my research. So today the talk is about how and why does soil organic carbon change from the perspective of biogeochemical controls. My research field is soil biogeochemistry. This talk will cover the main work in the past few years when I started my postdoc at Dr. Stephen J. Hall's lab. My main work is to study biogeochemical controls on soil organic matter dynamics in both natural and agricultural systems. I use the lab incubation, field measurement, and data model integration to understand how the interaction among plants, microbes, and minerals, particularly iron, controls soil carbon decomposition and the response to the agricultural management and climate change. You might have known the benefits of soil organic matter and its importance. As a large amount of carbon is stored as soil organic matter, small change in SOC can have big climate change. So science and the policymakers are very interested in building carbon to help mitigate climate change. However, the mechanistic controls on SOC stabilization are still controversial. Here I list three main important mechanisms for SOC protection. First, the biochemical stabilization. So carbon with more complex molecular structure is hard for microbial decomposition, so it can persist in soils. Second, the chemical stabilize. However, some scientists argue that the molecular composition might be not important. Second, for the chemical stabilization, this will occur because of the chemical and physical chemical binding between soil mineral components and carbon. So more binding means more protection. However, some minerals like iron is redox active. If the mineral phase change, then what would happen to the associated carbon? Third, less oxygen availability, less carbon decomposition. So oxygen limitation is an important mechanism for SOC protection in the sediments and wetland. This is because the kinetic and thermodynamic constraints. However, it is still unclear whether it is important in soils or upland soils that experience oxygen fluctuation. So based on this, I raised three main questions. First, does molecular composition influence SOC persistence? I will give you an example about the lignin fate in soils to answer this question. Second, is mineral-associated organic carbon necessarily stable? I will use an experiment with excess soil moisture to explore the geochemical controls on carbon decomposition. Third, is oxygen limitation important for SOC stabilization? I will use an oxygen fluctuation experiment to address this question. Finally, I will show you an example of field study on how soil moisture affects soil organic carbon. So let's look the first example about lignin fate in mineral soils. Lignin is one of the most abundant organic polymer on Earth. It comprising 10 to 30% of plant biomass. This is show how lignin chemical structure look like. Due to this complex structure, lignin decomposition is usually slow and represent a bottleneck for later stage of little decomposition. So plant little decomposition is the major carbon input for soil organic matter. In the litter, there's lignin and non-lignin. Because as lignin is hard for microbial decomposition, we would expect more lignin persisted in soil and contribute to SOC. So relative to the other carbon compound, lignin is usually thought to accumulate in soil, comprising major component of soil organic matter. However, recent studies have challenged this thought by showing that lignin actually can decompose faster than bulk soil organic carbon. The modern paradigm of SOC proposed little importance of molecular composition. Instead, it proposed the importance of ecosystem properties and microbial carbon use efficiency. A popular framework about the microbial carbon using efficiency is that it proposed the carbon from organic polymer like lignin with poor microbial carbon use efficient may be more likely to lost as CO2 than to contribute to the SOC. So as lignin, as a small fraction of lignin is typically assimilated by microbial biomass, we would expect lignin contribute less to SOC. Instead, the more labeled carbon component that are efficiently converted to microbial biomass and necromass tend to accumulate in soil and contribute to SOC persistence by interacting with minerals. However, we argue that this framework does not consider the impact of compound-specific interaction between carbon and minerals. We know minerals can protect SOC through binding. And many studies have shown that iron can preferentially associate with lignin and decomposition products relative to other carbon compounds. So the interaction of lignin decomposition product with iron may be an important mechanism for incorporation of lignin carbon to mineral-associated organic matter. So based on this, we hypothesize that lignin can be an important contributor to SOC due to the direct interaction with iron, and the importance of lignin varies with geochemical composition. So to test this hypothesis, I use a lab incubation and carbon-labeled lignin to track lignin fate in soils. Iron-rich tropical rainforest was chosen for this study. So after around 1 year incubation, we calculate the total decomposed carbon loss. And we use the proportion of total decomposed carbon loss to their initial added liter of lignin carbon. So in this figure, we can see that lignin carbon decomposition was much smaller than the liter carbon decomposition. We also use a density fractionation to see how much liter and lignin carbon remaining in the mineral associated organic matter. So we found that a great proportion of lignin carbon remaining in the mineral associated organic matter than did the liter carbon. So this evidence supports that lignin carbon can preferentially associated with the iron. Besides, we also collect soils from the 20 neon terrestrial sites, spanning from the tundra to tropic, to maximize the difference in geochemical properties and the microbial community composition. We examine the controls on lignin decomposition at the continental scale. We use the linear mix model to see which variable is important predictors for carbon decomposition. Today, let's just focus on the geochemical controls on lignin decomposition. So the red point and line represent the lignin decomposition incubated in the lab, and the black point and line represent lignin incubated in the field. And the x-axis represents the standardized coefficient, and the y is those important variables. So we can so the positive value means there's a positive relationship between the variable and the carbon decomposition. So we can see the reactive ion is an important negative predictor for lignin carbon decomposition. This is consistent with our previous results. So more iron, in that case, less lignin decomposition. That means more lignin was protected by the iron. Another interesting is that manganese is an important positive predictor for the lignin decomposition both in the lab and the field. This is because manganese is an important component of lignin degrading enzyme and also is an important catalyst for lignin decomposition. So this evidence supports that support the fact that lignin mineral interaction varies with geochemical composition. So to answer the first question, does molecular composition influence SOC persistence? The answer is potentially yes. Lignin is an important contributor to mineral-associated organic carbon, but lignin contribution varies as a function of ecosystem properties. So our study partially recognizes the idea from the classic and the modern SOC paradigms. As we know, mineral can protect iron. The next we would like to ask is this mineral-associated organic carbon necessarily stable? So I will use the experiment with excess soil moisture. Let's look at this cartoon first. The orange represents the oxidized iron, and the triangle represents the carbon. So under the aerobic condition, the minerals and iron combine the carbon and protect carbon from microbial attack. So and less carbon is available for microbial decomposition. However, iron is a redox reactive. And under the anaerobic condition, so iron, those oxidized iron can get reduced and become dissolved. At the same time, the iron bonded organic carbon can be released and become available for microbes. In this case, there will be more carbon available for microbial decomposition. If this is the case, the mineral-associated organic carbon may be not always stable. In the traditional view, High soil moisture, less oxygen availability, and correspondingly lower carbon decomposition. So based on this principle, soil carbon model usually assume decreased carbon decomposition under the high soil moisture. However, under the high soil moisture, there's anaerobic condition, and the ion can be reduced. The ion-bonded carbon can be released and probably expelled as methane and CO2 to offset oxygen limitation. So we hypothesize that carbon decomposition under the high soil moisture might not follow this pattern. So to test this hypothesis, we collect 3 monosols from different topographic positions and incubate the soil under the different soil moisture, including field capacity, the blue line in this figure, and the intermediate soil moisture, the green line in this figure, and the saturated soil, the red line in this figure. The dashed line in this figure represents the saturated soil was drained after around 3 months. So for this total around 5 months incubation, we can see at the beginning of the incubation, carbon decomposition was suppressed under the high soil moisture. But after that, higher soil moisture had the great carbon decomposition. By looking at the cumulative total carbon loss, we can see the cumulative total carbon decomposition was significantly higher under the higher soil moisture across the 3 soils. Why causing this higher carbon decomposition under higher soil moisture? So we can see under the high soil moisture, the red line here, there's ion reduction occurs. At the same time, dissolved organic carbon increases. So this means the accumulative DOC might have failed increases in the carbon decomposition under this higher soil moisture. We further see the relationship between carbon decomposition and the soil moisture. We select 3 time points of carbon decomposition, include at the beginning of this experiment and right before soil drain and at the end of incubation. So we can see at the beginning of this incubation, our data fit the traditional assumption, which shows lower carbon decomposition under the higher soil moisture. However, after that, our data did not follow this pattern by showing that high soil carbon decomposition under the high soil moisture. So this evidence suggests elevated moisture can stimulate carbon loss from mineral soils by releasing protect organic carbon. So ecosystem carbon model could potentially improve the capacity of prediction by linking soil moisture to ion carbon interaction and including methane production as well as CO2 in the transient wetland ecosystems. So we have evidence to show that mineral associated carbon is not necessarily stable. If mineral associated organic carbon is not stable under the anaerobic condition, then we have another question. Is ocean limitation important for SOC stabilization? So we are going to have an ocean fluctuation experiment. Why ocean fluctuation is important? So so from this map, we can see a great proportion of hydric soils in the United Land area experience periodic saturation, indicating the importance of ocean fluctuation. Even in the well-drained soils, soils can have an anostic microsize. Soil oxygen availability might also temporal dynamic. So this photo showing is for soil experiencing oxygen fluctuation. So we can clearly see that they are both oxidized iron and reduced iron. It's the reddish is for the oxidized iron and greenish for the reduced iron. So oxygen fluctuation drive ion redox cycling, which could potentially affect the associated carbon. In the traditional view, less oxygen, less carbon decomposition, and methane production is generally thought to be smaller under the oxygen fluctuation. So this assumption implied that periodic oxygen limitation decreases total carbon loss and climate change and climate impacts. However, the iron-bonded carbon can be released following iron reduction, and the previous research, including our work, have shown that oxygen fluctuation can also sustain methane production. So based on this, we hypothesize that oxygen fluctuation can lead to equal or great carbon loss. As methane has much higher global warming potential, so we would expect oxygen fluctuation can lead to much higher climate impacts. So to test this hypothesis, I choose two soil. One is from the agricultural soil in Ames here, and the other one is from the tropical rainforest soil. Before I show you the results, I would like to spend a little bit of time to talk about the experiment design here. So for the oxygen fluctuation, we totally have 5 treatments. Let's take the 48-day incubation as example. The x-axis represents the time, the day, and the y-axis represents the oxygen concentration. So 20% is represent the oxic condition, and 0 represent the anoxic condition. So for the control, the samples will incubated under the consistent 20% oxygen concentration, which is equal to the concentration in the atmosphere. And for a 2-day treatment, samples were incubated under anoxic for 2 days, followed by the 4-day oxic condition. And this oxygen fluctuation just repeated throughout this experiment. And for the 4-day treatment, sample will incubated under anoxic for the 4 days, followed by the 4-day oxic condition, and then just repeated this process. For the 8-day treatment, samples were incubated under the anoxic condition, followed by another 4-day oxic condition, and then repeated. For the 12-day treatment, sample will incubated under no oxygen condition for 12 days, and followed by oxygen condition for another 4 days, and then just repeated. Hopefully this will help you understand this treatment. Now let's look at the results. So let's look at the carbon decomposition first. So overall, both CO2 and methane fluctuate due to under the oxygen fluctuations. So different lines represent different treatment. And let's take the 12-day treatment during the 48 days incubation as example. So we can see, so remember that 12-day is the 12-day no oxygen, and followed by the 4-day oxygen. So under the anoxic condition, we can see CO2 production decrease and then increase under the oxic phase. But for the methane production, methane increase under anoxic condition and still continue during the oxic phase. So the similar results for another soils. So after more than 1 year incubation, we calculate the total carbon loss and based on the carbon isotope ratio data to separate total carbon loss into lethal carbon and soil carbon. So we found that oxygen filtration generally slightly suppress lethal carbon decomposition, but not for the soil carbon decomposition. And this result lead to very similar carbon decomposition between the 12-day treatment and the control. So after we convert all the methane and the CO2 to CO2 equivalent greenhouse gas emissions, we found that the 12-day treatment has much higher climate impact than the control. And the same pattern was observed in another soil. We further compare our data with model simulation. We use a microbial process carbon model. And first, we calibrate our model parameter with control only and apply transitional assumption. That is, carbon decomposition decrease with oxygen with lower oxygen availability. The blue line in this figure here. And the second, we calibrate our model with our full dataset. And the red line in this figure here. So the x-axis represents the cumulative oxygen availability relative to the control. And the y-axis is the cumulative carbon decomposition normalized to the control. So we can clearly see that the carbon model with transitional assumption totally failed to fit our data. And this the same case for another soil. So our data clearly show that so highest carbon loss from the oxygen limited soils, which change transitional model assumption. Based on this evidence, you might ask, is oxygen limitation not important for carbon stabilization? The answer actually is no, but perhaps it requires consistent oxygen limitation. In order to test the importance of consistent oxygen limitation, we have another experiment with static anoxic condition compared to the static oxic condition. So after more than 1 year incubation, we calculate total carbon loss and separate to lethal carbon and soil carbon. We found that anoxic suppressed total carbon loss, mainly due to the decreases in lethal carbon decomposition. However, it sustained or even increased soil carbon. We also have extraction data showing that soil carbon loss was much higher under the anaerobic condition. Today, I'm not showing that, but all of these suggest that the released mineral-associated soil carbon likely contribute to the sustained soil carbon decomposition under the anoxic condition. So overall, there's trade-off between oxygen limitation and mineral protection. That is, decreased lethal carbon decomposition was offset by loss of mineral-associated soil carbon. We conclude that temporary oxygen limitation may accelerate total carbon loss, and consistent anoxic is perhaps key for SOC accumulation in wetland soils. The last but not least, we use the field experiment to examine how moisture affects SOC and may provide some insight for drainage management. As a majority of US corn belt is drained by subsurface tile, the impact of drainage on environmental performance has received much great attention. However, its impact on SOC in crop depression remains unclear. A popular idea is that drainage can increase carbon loss due to the soil aeration. However, in this figure shows that SOC in the drained and undrained treatment in different soil series. From left to the right, soils are well drained and moderate gent and poorly gent. So we can see in the well-drained soils, soil carbon is pretty similar between the drain and undrained treatment. This makes sense. Drainage did not increase more carbon loss under the well-drained soils, but for the moderate drained soils, there's higher SOC under the undrained soils. However, once in these poorly drained soils, soil carbon is not consistently higher under the undrained soils across the profile. Most of the time, SOC is pretty similar between the drained and undrained. So in these poorly drained soils, it seems like drainage did not increase carbon loss. In these poorly drained soils, even drain depression can be flooded temporarily. So that means soil experience ocean fluctuations. Remember, my study showed that ocean fluctuation can have a higher carbon loss. So other factors might override the impacts of soil aeration induced by drainage on SOC in this crop depression. So we hypothesize that higher SOC often observed in crop depression might be explained by soil erosion alone, not suppression in carbon decomposition. So we collect soil sample from 7 topographic transect in cone soybean rotation in Central Iowa. So for each transect, there's 10 plot from the depression bottom to the upland. Let's look at the SOC and fine soil particle first. So we use the elevation relative to the elevation of depression boundary. So the plots on the left side of the dashed line represent the sample collected from the depression, and the point on the right side of the dashed line represent the sample collected from the upland. So we can see the SOC was higher in the depression than the upland. And this is consistent with the pattern of the fine soil particle. So these data suggest that soil erosion transports the fine soil particle and associated SOC from upland to the depression. For the soil CN ratio, if there's suppression decomposition in the depression, we would expect higher CN ratios. This is because the accumulation of carbon-rich plant materials. However, the soil CN ratio is pretty similar between the depression and upland. So ocean fluctuation in this depression might have sustained the carbon decomposition. So we further explored the relationship between SOC with the depression and the pounding. The x-axis is the percentage of pounding time in a year, which is averaged from 2017 and 2018. The pounding data was provided by Dr. Cater's lab, and each point represents the average samples from plots and depths with a pressure. So we can see that the pounding can vary from 5% to 50%, but the SOC did not change a lot. Even we can see more pounding seems to have a slightly less SOC. We, by using Lignin mix model to see which predictors are important for SOC change. So we can see the silt plus clay, the fine soil particle is an important positive predictor for SOC. That means more fine soil particle and higher SOC. And interesting, we found the reactive ion and the pounding is a negative predictors for SOC. Previous research have suggested that under the redox fluctuation, iron can serve as a catalyst for carbon decomposition. And more ponding, less SOC. This is my related to the suppressed plant growth under the ponding, therefore less carbon inputs to the soils. So overall, this suggests that soil erosion mainly drive SOC accumulation in these crop depressions. So we conclude that high SOC often observed in the crop depression was induced by soil erosion, not by anaerobic suppression of decomposition. And more poorly drained depression did not have great SOC. And increased drainage may not necessarily lead to more carbon loss. Instead, planting of flood-tolerant vegetation might help adding more carbon to this crop depression. The take-home point is that molecular composition can be important for SOC persistence, but this varies with ecosystem properties. Mineral-associated carbon is not necessarily stable under the redox fluctuation. Consistent anosmic is perhaps key for SOC accumulation. And the high soil moisture does not necessarily result higher soil carbon. Finally, I would like to thank to my supervisor, Dr. Stephen J. Hall, and ISU colleague, friends, collaborators, external cooperators, and undergrad students. Thank you. Any question? Yeah.
Question for one.
Yes.
So ask, where did the oxazole come from?
Yeah, so I forgot to mention. So oxo is from the tropical rainforest from Puerto Rico. Yeah, we collect the soil and then ship the soil to our lab to do the lab incubation. Yeah. Good point. Yeah, I should mention that.
Didn't come from Iowa, huh?
Because that soil is iron-rich soil. So yes, Marshall.
I've got a question. Thanks for the presentation, by the way. I really enjoyed it. I liked all the different kind of chapters you had in there. I'm really interested in hearing you elaborate more on the idea of the tile drainage and the losses of soil organic carbon. You alluded to that study, they're quite variable. The Fenton, I think it was a Fenton.
Yeah.
But would you care to speculate on why we've seen, up to at least 50% or more decline in soil organic carbon since cultivation, 150, 250 years ago in the Midwest? It's not tile drainage. What do you think the primary cause is for the loss of soil organic carbon observed compared to native grasslands?
Yeah. Oh, thank you. Yeah, this is a good question. So about why we observe soil carbon decline after we converted the native prairie to the cropland. So yeah. So first of all, I think the tillage is the important factor drive SOC decline. And the native prairie usually have more, I think, have kind of more consistent saturate. In that case, the probably oxygen limitation also contribute to protect the carbon. Yeah, but after converted to cropland, probably more soil aeration and also the tillage to disrupt the physical protection. Yeah, so cause the soil carbon decline. This is my thought. Hopefully.
I do have a question from online.
Yeah.
Thanking you for a great presentation, and then mentioning in the second experiment consisting of the MOAM stability, were those soils used for incubation oxasols? Would we expect similar results for mollisols?
So which you mean the first experiment? Oh, sorry.
She said the
Second experiment?
Second experiment about excess soil moisture. So actually, we use the oxic. Let me see. We use the mollisol. Oh, sorry. Yeah, this one. Actually, we use the mollisol in M's here. So the question is that whether it is will also apply to the oxo. Is that right? Oh, so so we have no idea. So actually, our data reveal the mechanism why cause the higher carbon loss under the high soil moisture. At the same time, we also for the following following that we also have include the oxo, both oxo and mollisol to do this experiment under the pretty similar mechanism, and to see, because under the redox fluctuation, the mineral bounded carbon can be released and become available for microbial decomposition. So that's why it causes the high carbon loss. So in that case, I would expect, so it will be the same similar results for the oxo, even we just did a mollisol. Yeah. Thank you. Yeah, good point. But it also deserves our further research to see whether this is consistent across a broader scale. Good point.
I will also ask one more question.
Yes.
So just on this slide, you explain your experimental design for using different topography here.
Oh, this one. Yeah, yeah, good, very good point. The reason is that because different topography have a different gend characteristic. So we want to see whether this is only occurred in the poorly drained soils, or also actually also apply to the well drained, so well drained or moderate drained. So the ridge is represent the well drained soil, and the foot slope represent the moderate drained soil, and the depression means the poorly drained soil. But we found the consistent results across the three soil.
Moisture is more important than topography.
Yes. Yeah, because the same mechanism applied to these three soils. Thank you.
Any further questions?
Nothing else is coming online.
Did I stop sharing? I forgot.
No, you're still sharing.
Oh, okay. Yes, sorry about that. Yeah. Thank you so much. Yeah, thank you. Thank you again for your invitation. Yeah. Okay. Oh, I should stop.
You're getting some people clapping for you online too.
I stop.